And in this past paper question, you're being tested on the topics of cell structure and function, as well as the characteristics of life. So the first part of the question, you are presented with a diagram. And as it says here, figure 2 shows a typical plant cell. Let's zoom in a little bit. Right. So a very good strategy that I tell students is anytime you are presented with a diagram, go through the diagram. Make sure you understand what's going on. Make sure you understand the various parts of it before you even go on to the rest of the question. Because interestingly, by the time you go through the diagram, analyze it, retrieve whatever information you remember based on it, you might have already answered several of the questions coming up just by doing that. So let's go through this diagram. So you'll notice that it's the question says that the, the diagram is showing you a typical plant cell, but even when we look at the title, it just says a typical plant cell. It does not indicate whether it's a typical plant cell as seen with a light microscope versus as seen with an electron microscope. So that is something we need to pay attention to, because now we have to ask ourselves, okay, based on what I'm seeing here, does this appear to be a plant cell as seen with a light versus an electron microscope? And two things that would two observations would tell you that it's as seen with an electron microscope. And it's these structures here. So you see these membrane systems, you would not see them with a light microscope. So you can definitely say that this is a plant cell as seen with an electron microscope. So that's the first thing we make note to ourselves about. And what I'm doing right here is something you can do during the exam or elsewhere, just generally analyzing something, you know, interpreting some information in front of you. You jot down, take a pencil and jot down some information as it comes to you based on the diagram. So the other thing we'll notice is that even though there are several structures presented, as you would see in a plant cell, only four of those structures are labeled, and they are labeled with letters, right? So one of the first things you can do is write down what those various labels are. Okay, so A would be the nucleus. It's the second largest um, organelle, and its overall structure tells you it's a nucleus, including you have this dense structure within the nucleus, which is called a nucleolus, which you may come across every now and again, right? It's definitely not a chloroplast, which is the third largest organelle. It's slightly smaller than the nucleus. So we know that C, which is pointing at this particular structure, we know this one is the chloroplast, right? And the next structure, let's go to B. So B is a membrane system, and the membrane system that we talk about at this level would be your endoplasmic reticulum. And some students would include the detail of it being a rough endoplasmic reticulum. Either way, you are fine, you are good to go, because yes, there is a part of it that shows some ribosomes embedded on it, but it's not the entire thing. So either way, you're okay. If you say with or without indicating that it's a rough endoplasmic reticulum, you will still get it correct if you answer it this way, right? The next one is D, which is the mitochondrion. right? Because we can tell it's a lot smaller and it has that zigzag shape inside of it, even though it's a little more detailed as what you would see in a light microscope. So you can let's look at this one here, right? It has that zigzag arrangement. So I have been asked, how do you tell the difference between the two in terms of the lines, the linear structures on the inside? So the chloroplast, you'd notice that the linear structures would be along the length of the organelle, right? So it's along the length. And the mitochondrion, that zigzag arrangement, the lines, you see lines going across the width of the organelle. All right. So if you're really lost and you're really not sure, look for that. Where are the lines across the length, along the length of it, or across the width of it? If it's across the width of it, then you're looking at your mitochondrion. If it's along the length, then you're looking at your chloroplast, right? So that's a little tip you can use to distinguish one from the other. So other than that, we are good to go. One thing I have seen some students in would name this the Golgi. Now at CSEC level, the Golgi complex isn't dealt with in much detail because there would be very specific features you'd look for to distinguish the Golgi complex from the endoplasmic reticulum. So save this for CAPE, right? This is a CAPE level detail, right? Advanced level detail. Because what can happen if you try to go beyond, you will end up naming an endoplasmic reticulum a Golgi complex and vice versa. And the Golgi complex has a very specific arrangement. Yes, it's a membrane system, just like the endoplasmic reticulum, but it has a very specific arrangement that tells you, okay, this is a Golgi complex, right? So avoid this, please. Avoid. Okay, at CSEC level. We're not dealing with that. Okay? So it's easy for you, don't make things more complicated than it needs to be. Once you see a membrane system, you know the only membrane system you're dealing with at CSEC level is your endoplasmic reticulum. And leave it at that. Okay? Good. So let's go to the questions now. So we've gone through the diagram and we've basically answered part one already, right? A, which is this guy up here, that's the nucleus. B is your endoplasmic reticulum. So we've already marked these two, right? We've already answered those two, so we just write it in, okay? Part two, you'll notice that it says the function. So if you answer this part by just naming the organelle, you still are not going to get the marks because they were not asking for the name of the organelle. They were asking for the function of the organelle. All right. And I have seen a lot of people writing a lot of very fancy descriptions. All right. Keep it simple, right? Chloroplast, site for photosynthesis, or photosynthesis occurs here. Mitochondrion, site for aerobic respiration, or aerobic respiration occurs here. On several occasions, I've seen some students giving very long descriptions for mitochondrion. It's the place where energy is converted to ATP and energy is converted to chemical energy for the cell to use, and la la la, and still don't even name the process. Name the process, right? Even if you want to go into a long description of what it is, still give us the name of it. All right. There was there's one example of someone indicating that the chloroplast is where light energy is trapped for photosynthesis. And yes, that is part of what the chloroplast does. The part of the chloroplast that traps the light energy is your chlorophyll, which is the pigment. right? So the chlorophyll, right, that's what gives the green color, right? That's your green pigment. I'm saying green. Why am I saying green? The green pigment, right? That would be in the chloroplast. That is what that's what traps the light energy. And then you'd have other parts of the chloroplast that would then use the light energy to carry out the process of photosynthesis. So the chlorophyll is the pigment, the chloroplast is the organelle, the source of energy that's used to run the process of photosynthesis is light, and I should say, let me rephrase, the type of energy that's used to run the process of photosynthesis is light energy. But the entire process, which includes the conversion of carbon dioxide to a carbohydrate, namely glucose, that entire process is photosynthesis. Okay, so make sure you can distinguish one from the other. Okay, and then most people answered this one correctly in terms of what is the feature that's common to both plant and virus, plant cell and virus. One or two people said cytoplasm. Cytoplasm, viruses do not have cytoplasm. It's the one microbe that does not have cytoplasm. Every other microbe has cytoplasm. Okay? And for this one now, differences that would be observed between the virus and the typical plant cell. I found a lot of people clearly were doing some Googling and giving some really in depth, detailed answers about the difference between a virus and a plant cell and still not answering the question, because the question started off with this statement. It started off with this sentence: you're required to view a virus and a typical plant cell under a microscope. Now, already you know it's going to be an electron microscope. Why? Because you're viewing a virus. Okay? Most viruses cannot be seen very clearly with a light microscope. So you're looking at samples of a virus and a typical plant cell under a microscope, which we figure out is electron microscope. And if you're using an electron microscope, the samples are going to be dead samples. They're not going to be live samples, which means that you are essentially looking at two static images of a virus and a typical plant cell. So, in the context of that, you're going to give differences. You're being asked to suggest differences that would be observed in those two static images. Okay, so you're not talking about the virus life cycle or how often this plant cell reproduces, etc., because that's not the information you're going to get from static images. Okay, the information you can get would be the physical features of the virus versus the typical plant cell. So you would include things like, the virus doesn't have a cell wall. I put cellulose in brackets because, again, you're not really going to see if the cell wall is made of cellulose. At best, you may be able to see that the cell wall is made up of these fibers, right? But you're not going to see that it's a cellulose cell wall, but you will see that there is a distinct cell wall around the typical plant cell versus a virus that's not going to have a cell wall. It's going to have this protein shell. Okay, so that's one difference. A plant cell has a cell wall, a virus does not. And then you can literally go down the line in terms of distinguishing. You can talk about the chloroplast, nucleus, mitochondrion, any of those organelles that you would find in a plant cell that would then be absent in the virus. And those all of those differences can be used to distinguish between the two, or that you'll be able to see between the two. All right. Please avoid shape, avoid size comparisons. All right. The first one, why? Because there is a wide diversity of shapes that you would find in plant cells. And yes, it's a typical plant cell, but again, there's also a wide variety of shapes that you find in viruses. Okay, so you don't really want to use shape to distinguish between the two. All right, only if you want to talk about the overall structure, that the plant cell has a cellular structure where you have a membrane and cytoplasm and organelles. You can even say that. And then you can say, well, the virus has a very different type of structure where you have this protein shell with some DNA or RNA on the inside of it, with some kind of genetic material on the inside. Okay, you can even talk about the lack of um, extra structures that you find within a virus as compared to a plant cell. Even that's a difference, right? That the plant cell has several sub structures within it, right? Like all these organelles that you would not find in a virus. So avoid shape, all right? If you want to talk about the overall arrangement of the organism, fine. But don't say, well, this one is round and that one is rectangular. No, try to avoid that. Okay. Size comparisons, again, because you're not being given scale, etc. You really want to avoid size comparisons. All right. The only time you could use a size comparison is if, for example, they give you two actual micrographs and they indicate the scale of each one. So they say, well, this one is magnified 10,000 times and this one is magnified 100 times. Then you could compare the size. Only if you are given a clear scale or magnification, then you could talk about the size, compare the size of the two. There was no such indication here. You just were told that you looked at it through a microscope. All right, so no size comparisons, um because again, the point is you're talking about differences that would be observed. You're not talking about differences generally. You're talking about differences that would be observed in this given context, that you're looking at it through a microscope. Okay, so again, with these two, don't skip over that first sentence in these types of questions. Always read that first sentence, that scenario. Pay close attention to that scenario. Okay? Because a lot of times that's how people miss the marks. They write a wonderful answer, are convinced that they wrote an amazing answer, and completely ignored the scenario that was presented. And then they just answered based on background knowledge and not based on what the situation was. Okay, and that's also an important strategy for life, right? Okay, pay attention to what's in front of you. All right. For the for this question, why is the neuron, nerve cell, straightforward, differentiated? Right, these are all differentiated cells, right? W might be considered undifferentiated, but because we want to talk about all three, they didn't really say one versus the other. We can indicate that, well, the majority of them are differentiated. X and Y are definitely differentiated. W might be some kind of epithelium or some other kind of cell that may or may not be. But again, the majority of them are. And for those of us that forgot, saying that something is differentiated is the same as saying that it is specialized. Okay? Undifferentiated would, of course, be the opposite. It would be unspecialized. Okay? And then for this one, again, it's straightforward. You want to pay attention to the scenario presented, right? A lot of people, well, there were several persons who just answered the question based on, well, I know the characteristics of life, so just pick three. No, you have to answer given the scenario. All right. So based on the scenario, these are the characteristics of life that were being illustrated while they were playing in the park. All right. Movement, they were running around, breathing deeply, respiration, laughing when they're hurt, running to mommy for a snack, nutrition, response to stimuli. All right. Some people indicated reproduction with a stretch, yes, but it's not a straightforward connection, right? If, for example, they had said they run to mommy for a snack and mommy is pregnant and she's watching over the children and she's pregnant, then yeah, that's definitely going to illustrate reproduction, right? Some people illustrate said it illustrates growth. Not directly, it's not a direct illustration. Um, an illustration of growth would be the children in the preschool were running around the park, and 4 or 5 years later they no longer play in the park because they've gotten bigger, or they've gone to a new school, right? Because they're now a lot taller and they're bigger and they're doing other games, right? As 5 years later they're now playing football in the park. Then it's like, oh well, yeah, that illustrates growth because time has passed and they're showing that the children have grown and developed over time. All right. So that's how you make the connection between a scenario and what does it illustrate. You always look for the most direct illustrations for these kinds of situations. Okay. And for the last part, right? So we're talking about the Georgiana saying, sorry, Bernadette is saying that the flame is a living organism, and she's giving three characteristics, three features that to her demonstrates that the flame is a living organism. And to answer that one, it's a very straightforward answer. To any body, any object, any anything that you're observing or investigating to figure out whether it's alive, it's a living organism or not, it's a living thing or not, you have that thing has to clearly demonstrate at least seven characteristics of life, which we use the acronym GRIMNA to list, right? In this case, Bernadette only gave three. So even if, let's say, it really did move like a living organism and it did grow and reproduce like a living organism, it's still just three features. It's still just three characteristics. So it still fell below the requirement, right? So that in a question like this, you keep it simple. Said, well, you need to cover seven, only three was described, so therefore that alone tells you it's not a living organism, right? You will be coming across other types of questions where you have to now go into detail about defining the characteristics and showing how a particular feature of some non-living thing does not fit that characteristic. So, for example, they may say, "Well, a car moves, so therefore um, it is demonstrating the characteristic of movement." And you would have to say, "Okay, yes, but it is not moving on its own volition. It is the only way it moves is if you sit in the car and you make it move, right?" So therefore, it does not really fall under the definition of movement for a living organism. So therefore, you cannot use that to say it's a living organism. So that's another kind of question you may get. You would get. But that's basically it, right, for this particular paper. And I look forward to seeing how you all deal with the question coming up. Okay.